In this video, we're going to take a look at the general structure of membrane lipids. Now, I've arranged membrane lipids into these three categories. There's different ways of doing this. This is just the way that it works in my head. Now, the membrane lipids, we break them into phospholipids, glycolipids, which have somewhat similar structures, and then there's something completely different, and that's cholesterol. And so we're going to take a look at cholesterol separate because it is just structurally so different. But now the phospholipids and glycolipids, if we just focus on those for right now, these membrane lipids have two basic parts to them. They have a head and a tail. And the head on these are polar. These are polar, and then the tails are nonpolar. So if we were to kind of diagrammatically draw out one of these membrane lipids, we would have a polar head like this, and then we would have tails coming off of it, two tails. And the tails here, kind of zigzaggy lines like this, like that. Okay, so then I'm just going to say up here polar and non-polar. Like that. Now, these phospholipids all contain a phosphate group. So I'm just going to put in here phosphate group. So I'm just going to say contain phosphate. That's just a P with a circle there, group. Contain a phosphate group. And the backbone of this first one here, this is a glycerophospholipid. Glycerophospholipid. And the backbone of this is glycerol. Glycerol. And then attached to the glycerol are two fatty acids. And the fatty acids are nonpolar. And these are attached with ester linkages. Now down here, we're going to have a polar phosphate group. A polar phosphate group and an alcohol. Now I'm just going to write in an alcohol group here to show that it's an alcohol, but it could be a number of carbons just ending with an OH, so an alcohol group. So that would make this portion here polar. So this would be the polar head, and these here would be the two nonpolar tails. Then if we take a look at sphingolipids, sphingolipids here are going to have this big long backbone, and this big long backbone kind of folds around over this way and doesn't leave room for three groups to attach like this. So there's only going to be two groups attached. And so this sphingosine, let's see if I can write that in there. Sphingosine. Sphingosine. Kind of goes around like this, and then we're going to have a fatty acid attached. Fatty acid attached. And then we're going to have a polar phosphate group and an alcohol. And again, the alcohol here could be a number of carbons attached just as long as it ends with an OH or has an OH attached to it. And these, again, would be ester linkages. So this part of the molecule would be the polar head. And then here we have the two polar tails. And part of the tail here is the sphingosine molecule. Then over here we have glycolipids. Now glycolipids, the name glyco means that there's a carbohydrate involved. So contains carbohydrates. Contains a carbohydrate, and we're going to have the same backbone like we had here, the sphingosine. and our fatty acid. And then attached, what makes it different is going to be that carbohydrate. Carbohydrate. And again, these linkages here, these are ester linkages. Ester linkages, ester linkages, all attached onto the backbone. All right, so that's two of the three categories of our lipid membranes. The next one we're going to take a look at is cholesterol. The third type of membrane lipid we're going to take a look at is cholesterol. Now, cholesterol, there's good cholesterol, there's bad cholesterol. You may have heard of these, and we do need some cholesterol. Cholesterol is a lipid, it's in lipid membranes, and it helps to keep the lipid membranes nice and flexible. So if you don't have cholesterol in there, those cell membranes can't flow the way that they're supposed to, and maybe those cells can't get into the spaces that they need to. So the basic structure for cholesterol starts with this. This is what we call the steroid nucleus. And the reason it's called a steroid nucleus is because we're going to see a lot of other structures that start out with this, and then we just add and remove different pieces to it. But this is like the basic structure for cholesterol. And so if we're going to turn a steroid nucleus into cholesterol, we need to add a few things. So we're going to add a hydroxyl group here, like this, and then we'll throw in a methyl group, like that, and we'll put another one over here. And then here we're going to have, let's see, 6 carbons coming up here. 1, 2, 3, 4, 5, 6, and then a little branch coming off of there, and a little branch coming off of there, like that, kind of running out of space just a little bit. Now, don't worry about like having to be able to draw this on a test or something. I'm not going to ask you to memorize this structure, but when we add these things to the steroid nucleus, now it becomes cholesterol. And cholesterol, as you look at this, it doesn't have a whole lot of strongly electronegative elements on there. I mean, there's an OH group over here, but this is primarily nonpolar. And so I'm just going to say here, cholesterol, let's see, it's nonpolar. And because it's nonpolar, it doesn't dissolve in the blood, but we need it to get to different parts of the body through the bloodstream. So the way that we make it soluble in blood is we attach the cholesterol onto some proteins, and these are called lipoproteins. Lipoproteins. And there's two basic types of lipoproteins. There's LDL and there's HDL. And what this stands for is low density lipoprotein, and this is high density lipoprotein. And the basic functions of these is these are going to be transporting cholesterol throughout the body. Now the LDL, low density lipoprotein, this is going to take cholesterol from the liver to the tissues. From the liver to the tissues. And HDL is going to do the opposite of that. It's going to take cholesterol from the tissues to the liver. And the way that I think of this is HDL is kind of like the garbage truck of the cardiovascular system. It's just kind of cruising around. It sees garbage like cholesterol lying around. It just scoops that stuff up, brings it back to the liver, so that way it can be metabolized. Now these two lipoproteins, the LDL is the one that we think of as being the bad cholesterol, and the HDL is the good cholesterol. Now, at your age, generally, as a typical college student, you probably don't care or even think about what your cholesterol level is. But as you get older, this becomes a number that your doctor makes you painfully aware of. And so they say, well, here's your number of good cholesterol, here's your number for your bad cholesterol. And you want to have your good cholesterol higher and your LDL, your bad cholesterol, lower. Because there is an association between having high LDL and cardiovascular risk, and having high HDL and lower cardiovascular risk. It's almost as if these garbage trucks are picking up cholesterol and cleaning it out of your cardiovascular system. So you want to have lots of this, not so much of this stuff here. And you're like, okay, so how do I fix that? Okay, how do I prevent myself from having high cholesterol? Well, cholesterol is produced primarily by animals. And so if you were to go to a non-animal diet, then you would have virtually no cholesterol in your diet, all right? And what does your body do in response? Well, your body still needs cholesterol for its cell membranes, so it will produce its own cholesterol. So it'll start producing LDL to kick it out to the tissues. But there would be less. And so people that have high cholesterol, one of the things they tell them is to cut down on the animal products in your diet and increase the fruits and vegetables. Because fruits and vegetables, I don't want to say they don't produce cholesterol, but they don't really produce cholesterol. I mean, it's just very, very small amounts. So I'm not trying to tell people to be a vegetarian. I mean, I love me a good carcinoburger once in a while, but for health reasons anyway, limiting the amount of animal products that we consume has been shown to lower our cardiovascular risk.
Hey! I saved your life. That egg sandwich could have killed you by cholesterol.
Forget it, Homer. While it has been established that eggs contain cholesterol, it has not yet been proven conclusively that they actually raise the level of serum cholesterol in the human bloodstream.
So one of those egg council creeps got to you too, huh?
Oh, you got it all wrong, Homer. It's not like that.
You'd better run, egg!
Welcome back, biochemistry students. In this video, what we're going to take a look at is the hydrolysis of a phospholipid under acidic conditions. Now, it seems like a lot of gobbledygook, but we'll work our way through this. Now, to get us started, this right here is a phospholipid. Now, this looks very similar to a triglyceride, which we saw in a previous video. A triglyceride is where we have this glycerol structure here, the 3 carbons, and then attached to the 3 carbons, or the glycerol, would be these fatty acids attached through ester linkages. Okay? Now the only difference is this time we have a phosphate group attached. That's just what makes it different. Now the ester linkages, if we remember what an ester is where we have the carbonyl group, and then the carbonyl group has a carbon chain coming off of it, and then here we have an oxygen, and then another carbon. So this right here would be a carbon ester group, and then a phosphoester group would be, instead of a carbonyl, we have a phosphorus double bonded to an oxygen, and then over here we have an oxygen attached and then a carbon. Okay, so this here would be a phosphate ester, and then on this side as well, we would also have a phosphate ester. So there's 2 of them here because we have the carbon chain coming off of this oxygen and a carbon coming off of that oxygen. Okay, so just kind of getting the lay of the land here, what's going on. Now this reaction, what we're going to do is we're going to react this here with some water. So plus H2O, and in fact, I'm going to rewrite this. I'm going to write this as HOH, so that way we can see that it breaks apart. So HOH, and then we're also going to be reacting this under acidic conditions. So we might say HCl might be an acid, or we could also write it as H+, okay, just acidic conditions. And what we're going to do is we're going to break apart these water molecules like this, and we're going to break these ester linkages and just break this whole thing apart. Okay, so now I'm going to rewrite this as we're breaking this apart. Now the last video that we had, we saw that we could break these apart and form fatty acids. Okay, so we're going to break this apart and form fatty acids. We're just going to break it right here, and then we're going to end up with a fatty acid up on top. Okay, so that is going to give us here, let's see, a oxygen, and then double bond O, carbon. Whoa. Let me rewrite that there. Okay, that's going to give us a carbon, double bond oxygen, and OH like this, and then our carbon chain coming off like that. We're going to have another one here, and so that's going to be OH. Let's see here, carbon, double bond O, and then, okay. All right, so those are our two fatty acid groups there. So I'm just going to label these two fatty acids. Okay, that's two of our products. And then we're also going to end up here breaking this apart. Now when we break apart our phosphate diester, there's two ester linkages here. We're going to end up here with our phosphate group forming OH bonds, just like we did here. Okay, so that's going to give us our phosphate here, phosphate, double bond, oxygen. Okay, just like that. So that's this right here. And then we're adding water to it, so we're going to break apart this bond here, and we'll end up with OH and OH like this. What we'll end up doing is breaking these bonds here, and then we end up with this molecule here. Now, I think I'm missing an OH up here, an OH here. Okay, and so this right here becomes phosphoric acid. Phosphoric acid. And phosphoric acid has a formula that's H3PO4. Not that you need to memorize that, but you might see that in your textbook. Okay, so here's phosphoric acid, and then what happens to this linkage right here? Well, this ends up becoming an alcohol. Okay, so we ended up with an OH coming off of this right here, and then so this gets broken apart and we end up with an alcohol. So I'm going to go like this and go OH like this, and some carbons like that, and that's going to be some alcohol. An alcohol there. And then the one thing that we're missing is our backbone here. This right here is our glycerol backbone, and so we're going to end up with 3 carbons with 3 hydroxyl groups coming off of that. And so if I were to draw that, we'll do it here. I'm going to go carbon to OH like this, H, and then carbon H2, OH, and then OH like that. Okay, and this is our glycerol group, group. So G, glycerol. Now, if I were to count up all of the waters that are involved in this reaction, I'm breaking bonds here, 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 and here. So this, if I were to balance this, I should put a 4 here. There should be 4 waters. There should be 4 waters there to balance this out. Okay, hope that helps. Hey biochemistry students, for this example what we're going to do is a very similar reaction to the last example, except the difference this time is we're going to be using a strong base instead of a strong acid. So everything else is the same. We start out with a phospholipid, but we're going to be using then a strong base. So our strong base is going to be sodium hydroxide, NaOH, and we're going to be using water. HOH. And just like what we did in the last problem is we're going to be breaking these ester linkages. Okay, so we have an ester linkage here where we have the carbonyl group double bond O and the oxygen. This is one carbonyl group. And here's another carbonyl group. And then here we have another ester linkage. In fact, we have two of them. We have an ester linkage here, like this. If we were to draw this out, here's one ester linkage. And then we have another ester linkage right here, like this, like that. Double bond O and then the oxygen and then carbons coming off of that. So we call this a diester. So this right here is a diester, and this right here would be a single ester and a single ester, like that. Okay, so the reaction is very similar to what we did last time, and that is we're going to take our water molecules here, we're going to break them apart like this, and then we're going to add them to our ester linkages here and then pull them apart. So I'm going to start out here with our ester linkages here that would form fatty acids. So that looks very similar to what we did before, okay, and that we're going to end up with our carbon like this, our carbon double bond oxygen like that, and then here is our carbon chain, and then we would have an oxygen. And I'm not going to put an H there just yet. So I'm going to redraw this, so I'm going to have two of them here. I'm going to go carbon, double bond oxygen, zigzaggy line like this, and then here is like that. Okay, now what's going to happen here is going to be a little bit different, a little bit odd, and that is we're going to take our water here, we're going to split this apart here, and this gives us sodium like this, sodium ions. And then those sodium ions, because they're electropositive, are going to be attracted to these electronegative oxygen atoms. So when that happens, we'll end up with sodium here, Na plus, Na plus, like this. And then this, instead of being called a fatty acid, it becomes a fatty acid salt. So I'm going to say 2 fatty acid salts. 2 fatty acid salts. And these have a fancy name. They're also called soap. Okay, and so soap is where we take a phospholipid like this and we break it down using a strong base. Okay, so that is soap. And then the rest of this is going to look fairly similar to what we did before. And that is we're going to end up taking this phosphate ester, this phosphate diester here, and then pulling apart the linkages. Okay, so I'm going to put in my phosphate here. So here's my phosphate, like this, double bond O, like that, like that, like that, like that. Okay, now in these very basic conditions, under very basic conditions like this, the hydrogens do not end up getting attached like this. And so we end up then with these just being like that. And you might be going, what? What's up with that? Okay, so here's the deal. We had, when we pulled this apart here, we ended up with sodiums going up here. And then we ended up with excess hydroxides here, excess OHs. Okay, and those excess OHs, I'm just going to put those down here. Those excess OHs, OH like this, that would take the hydrogen from here. Okay, so I'm going to put like a hydrogen here, and that's going to come down here, attached to this, and then we end up with HOH. Okay, so that ends up giving us equals HOH, which is, of course, water. Okay, so maybe I'll just go like this. So this plus this equals water. Okay, and so that's why this phosphate group here, like this ends up with just the oxygens on either end there. Those hydrogens would get removed. Okay, and then what do we end up left with then? Okay, so we have this chain right here. That chain right there ends up becoming an alcohol, and so that would end up here being OH, and then a carbon chain like that. So we'll call this an alcohol. And then we also end up with our glycerol, the backbone here. So glycerol. So CH2, CH. I can do better than this. I can do better, I swear. Carbon, like this, CH, and then CH2, OH, OH, and OH. And this is our glycerol group. Glycerol. Okay, so all this right here becomes glycerol. Ba-ba. That's how we make soap.